This set of standardized protocols can be used to offer new insight into the Superior Ocular Sulcus, or SOS, a recently discovered structure of the developing eye. These protocols will allow any experimenter around the world to clearly visualize the SOS during zebrafish eye development.
Begin by placing heterozygous growth differentiation factor 6A male and female zebrafish on separate sides of a divider in a tank of dechlorinated water the evening before their pairing. The next morning, remove the divider and allow the fish to breed for no more than 30 minutes. At the end of the breeding period, collect the embryos into petri dishes containing E3 medium for their incubation in a 28.5-degree incubator. At 20 hours post-fertilization, replace the supernatant with E3 medium supplemented with 0.004% 1-phenyl-2-thiourea to prevent pigment production, and examine the embryos by light microscopy to confirm they are all at the appropriate developmental stage. Place the samples under a dissecting microscope. Discard any developmentally immature embryos and use fine forceps to gently pull apart the chorions. Visualize the SOS, which may appear as an indentation or line at the dorsal margin of the eye. Then, sort the embryos that show SOS closure delay from those that do not. To photograph these embryos using a dissecting microscope, prepare a petri dish containing 1% agarose in E3 medium, and lightly prick the center of the agarose to create a shallow hole into which the yolk of the embryo can be placed.